So mutations are going to be heritable changes in your DNA, meaning you can pass it off from parent to offspring. So mutations are usually going to lead to alterations in your DNA, within your genome, and usually that difference in your DNA is going to, carry over to the transcript or the or the mRNA, and that's going to eventually carry over into the protein potentially. And so, usually, due to mutations, you're going to get differences in the kind of protein that you're making. And if you get differences in that protein that you're making, you might get a different phenotype. So, most of our differences between us are different phenotypes. I would argue are due to mutations at some point. So mutations can occur in two types of cells. You can have mutations that happen in your somatic cells. So those are all going to be all of the asexually reproducing cells in your body, like your skin cells, your or your epithelial cells. And then you could also get mutations in your germline cells. So the mutations that happen in your germline cells are going to be the types of mutations that you're going to pass on to your offspring. If you get a mutation in one of your skin cells, for example, or an epithelial cell, you're not necessarily passing on that mutation to your offspring. However, if you modify your germline cells, which eventually turn into your gametes, that's what's going to affect the progression of one generation to the next, or you could say the heritability of the mutation. So we have two different types of mutations. We could get chromosomal mutations, which are going to alter the total number of chromosomes. And when we do that, that's going to lead to very, very large structural changes. We could also get point mutations, where we're mutating a single gene within a genome. All right, so let's talk about different types of mutations. So we can get substitution mutations, and kind of as the name implies, you are substituting one thing for another. You're substituting one nucleotide for another nucleotide. So in this case, let's look at the DNA here. Here's our DNA in our nucleus, in our genome. And we've got AAA, ATG, CTT, CTC. Now, let's say we accrue a substitution mutation. We have AAA, ATG, and then instead of having a C, we have a T. So we have TTT, CTC. And normally we would have, this UUU coding for phenylalanine, UAC coding for tyrosine, GAA coding for glutamine, and then GAG coding for glutamine. And so what happens when we get that substitution, so we change this part of our DNA, which leads to this change in our mRNA. And eventually, now we've made a different amino acid sequence, a different protein. We have phenylalanine, tyrosine, lysine instead. So that's a form of substitution reaction or substitution mutation. That's a substitution mutation. Those compared to these other two could be pretty benign. Now, these other two, this is where you're making complete overhauls, and you'll see what I mean. So insertion and deletion, as the name implies, in the case of insertion, what you're doing is you're inserting a nucleotide. You're not replacing anything. You're inserting a whole new nucleotide. So originally your DNA was AAA, ATG, CTT, CTC. Now we have AAA, ATG, and then we insert a whole new nucleotide right here. But what that does is it shifts everything forward. So instead of CTT, now we have CCT for this codon. But instead of it going from CTT to CTC, now you have CCT, now you have TCT, and so on. So everything after that point, everything after that insertion mutation is affected. So if this was coding for a gene that was, I don't know, useful or necessary for life, you're not going to be living after this mutation, or at least that cell is not going to be living after that mutation. Now, if this happens in a germline stem cell, oftentimes that could lead to, a non-viable gamete. And if it does lead to, let's say, for example, a viable gamete, which eventually gets fertilized or fertilizes something, well, the organism itself probably wouldn't be viable. And so that could probably lead to a miscarriage if we're dealing with, mammals. All right, so insertion is going to cause a frame shift. So deletion, as the name implies, you're deleting, you're taking a nucleotide out. And so just to not, make this explanation too tedious, you have this ATG here, and what you're doing is removing this A, and what happens is it shifts everything back. So every codon preceding that deletion is affected, therefore leading to different amino acids. All these things labeled red are the different amino acids that are different from the initial. So these are detrimental. Your frame shift mutations are detrimental to the organism. All right, so now let's talk about some subcategories, or you could say the effects of these mutations. So we can have a silent mutation. So a silent mutation, as the name implies, is going to be silent. If you remember when we were looking at the codon chart, there is some redundancy between, what, and we could actually go back. We can go back here. Here we have some redundancy, like glutamine, it could be coded by CAA or CAG. So let's say, your original sequence was CAA and you replace that A with a G. Well, it still codes for glutamine. Thank goodness. Your protein is not going to be affected, assuming that's a good protein. Okay, let's go back to this. So in this case, you've got ATG, you have GAA, and that codes for glutamine. And what happens, this is the exact example that I used. You go from GA, what are the chances? So you go from GAA to GAG, well, thank goodness, it codes for the same amino acid. You're unaffected. This is a silent mutation. So here I just say, because of the redundancy in the genetic code, a substitution does not always lead to a different amino acid, therefore a different protein. So this is a silent mutation. It has no effect on the protein. You're going to get this from substitution. You could also get this from, a frame shift mutation, but usually a frame shift mutation is going to lead to, maybe you'll get like a silent mutation, initially, but everything after that, it's going to be jacked up, and there's a lot more mutations that are probably going to arise from that. So the next one we have is missense mutation. So missense mutation, that's where you actually get the wrong amino acid from substitution. So still kind of, risky. It could change the three-dimensional structure of your protein. If it's in the active site, it could change the enzymatic activity of if the protein was coding for an enzyme, or if the protein was an enzyme. So you could kind of nullify certain genes, or you could enhance certain proteins. So not all mutations are necessarily bad. They could lead to, a higher efficiency or a higher synergy with other proteins, for example. So missense mutation, the original code is cat, looks like cat, which is histidine. And what happens is, instead of this alanine here, you have a C or a cytosine, and you go CCT, which codes for proline. So you get the incorrect amino acid, which would cause a malfunctioning protein. And in the case of sickle cell anemia, this is exactly what happens. You get a mutation in your hemoglobin protein, and that single point mutation leads to this cascade effect, causing your red blood cells to change in morphology and fold in a weird way, kind of making them look like a sickle, you could say. And so your red blood cells become sickle-shaped. We call it sickle cell anemia. They have trouble carrying oxygen, and they also more commonly form clots. So this comes from a single point mutation. So here is just showing, the genetics of it. You could have an unaffected father carrier. So he has a normal allele and an affected allele, and he mates with a unaffected carrier mother, which has the normal allele and the affected allele, you could say. And so when you do their Punnett square, you have a 25% chance of making a child who is completely unaffected and a non-carrier. You have a 50% chance of creating a carrier child, and then you have a 25% chance of creating someone that is completely affected by sickle cell anemia. All right, so next we have nonsense mutations. So this is actually where you get a short non-functional protein. So this is usually going to be from substitution. And what happens with a nonsense mutation is, with our previous mutation, with our missense mutation, we got a new amino acid, right? A new amino acid in that protein. Well, in a nonsense mutation, what happens is you get a stop codon. So you've gone from something coding for a amino acid to something coding for a stop codon, therefore causing an early stop. So nonsense mutations lead to an early stop codon, so you get a shortened non-functional protein. So it pretty much silences whatever gene you were trying to express in that cell. Now, again, if it happens in your germline stem cell or in your germline cells, this could be detrimental to the future organism potentially. But if this happens in a somatic cell, well, I mean, this could lead to cancer in that region, or cancer within that cell, or whatnot, or you could just say a dysfunctional cell if it's vital for its survival. Next, we have frameshift mutations, and we already talked about frameshift mutations. These are detrimental. So these mutations can occur from insertion or deletion, and usually this is going to lead to, maybe a series of different amino acids, or it could be, an early stop codon, for example, or some silent mutations. But it's going to be a collection of all of them, and especially if this happens like at the beginning of your gene, this is going to be a weird protein that is nothing like the original form. So frameshift mutations, detrimental. It shifts the frame. The names kind of describe what they do. Insertion inserts something, deletion deletes something. And so here is just a table showing the different mutations.